Hi everyone, today we're going to look into what a PCR test is. Now, you could have heard of a PCR test, maybe heard of the word somewhere when you're visiting the hospital or trying to get your COVID-19 tested. But it's so much more than what you may typically associate it with, which is, I suppose, sticking up a long stick up your nose and having that painful and sore experience. But it goes far beyond than that. In fact, PCR stands for Polymerase chain reaction. And it is essentially referring to the exponential multiplication of a sequence of DNA. Yes, at a exponential rate. And the reason why we're doing this is we're trying to essentially amplify the volume of the DNA sequence to make it detectable, to raise it to a standard or a level or concentration where we can detect it, or even to be able to manipulate it. So it's essentially trying to amplify the volume or the amount of the specific DNA sequence that we have extracted from a human. And PCR is essentially the sort of a machine wherein we do this polymerase chain reaction. And what happens in this machine in order to multiply a certain DNA sequence is quite interesting. Now, in this machine, a couple of ingredients are needed. First is the TOC polymerase. We'll refer to this later on as a very important part of PCR. Second, we need free nucleotides. Free nucleotides is basically a necessary component for all sorts of DNA multiplication reactions because free nucleotides essentially assemble in order to form DNA strands. But besides that, we also need primers. Now, primers are essentially just short sequences of DNA that kind of get this process of creating and assembling free nucleotides together in order to form a strand of DNA started, but we'll talk about that later. So first, let's talk about what actually happens from the start. Now, what happens in a PCR machine where we're trying to get the multiplication of certain DNA sequences? Well, what happens is we go through this process of denaturization, wherein we're trying to increase the temperature to a temperature that's so high that the hydrogen bonds between the DNA sequences, DNA strands, will break, and this will cause the creation of two separate DNA strands. So this is where we try to raise the temperature to 90 degrees, essentially. And we're trying to break the hydrogen bonds in DNA strand. So we're going to create two strands from that because the hydrogen bonds that used to be between the nucleotides of the two DNA strands would be broken. And as a result, we're going to get two separate DNA strands. That's what we're going to end up with in this process of denaturation because we're trying to increase the temperature to as high as possible. And that is going to create two separate strands. Now, next is this process of annealing. And what happens in this process is we essentially attach or bond these primers, which are short sequences of DNA, to each separated strand. And what it looks like is kind of like this. So as I mentioned earlier, primers are essentially just short sequences of DNA. And we're attaching them to the end of each strand. So what's it's going to look like is, obviously we're going to have the 5' to 3' orientation on one strand, 3' to 5' orientation on another strand. And we're going to start right here. So we attach these primers to each DNA strand that's separated, and we do so by forming hydrogen bonds. And the direction that the primers are going to kind of extend and elongate is in this direction. And that leads me to the next process of the PCR test, which is elongation. And in this process, we use these specific DNA polymerases called the TOC polymerases. And what these Talk polymerases do is they essentially gather all the free nucleotides together in easy words, and they assemble them, and they kind of really align them in order to elongate the DNA, complementary DNA strand that forms for each of the separated DNA strands. So I'm just going to visually show you how this is going to look like with one strand. So with the help of the Taq protein, we're going to go in this direction. And free nucleotides are kind of going to be assembled in this direction in order to create another complementary strand. And obviously we have our primer right here. And as you can see, because these are complementary DNA strands, we are going to have to go in this orientation. Yes. To explain to you a little bit more about what Talk proteins are essentially polymerases. Talk polymerases are essentially enzymes. And what they are thermostable enzymes. And they have been derived from the bacteria Thermos aquaticus, which are essentially bacteria that have been living in hot springs at very high temperatures. And given that these Talk polymerases can withstand high temperatures, this elongation process is going to take place at a temperature of 70 degrees Celsius. So we're going to elongate the sort of complementary DNA strand started by a primer, and we're going to essentially form two separate DNAs. One, each complementary strand formed by the help of the Talk polymerase that starts off from a primer. And from then on, we go off to this other process of having to repeat this in a process called cycle. And we repeat this cycle of essentially creating complementary DNA strands, starting off from primers, using Talk polymerases, using free nucleotides, etc., in order to create two DNAs from one DNA. We repeat that process for around 20 to 40 cycles. And the goal of this is we're trying to multiply the DNA population as much as possible. So at the end of the day, we are going to end up with much more target DNA sequences. And that is how the process of PCR essentially takes place. Thank you so much for watching.